So in section 2, we'll be looking at the development of lymphocyte diversity. We'll look at the maturation process in a little bit more detail for both B and T cells, but pretty much the chart that I had showed you in the previous section goes over it in pretty good detail. Draw a diagram illustrating how lymphocytes are capable of responding to nearly every or any antigen imaginable. Outline the process of clonal deletion and clonal selection, and then describe the structure of both B cell and T cell receptors. So the markers, just to revisit the markers on T cells, and this first section is really about differentiating between B cells and T cells. So on the surface you have CD3 markers that surround the T cell receptor, and they assist in binding. The CD4 and CD8 co-receptors also help assist in a better fit. The CD4 also defines the type of cell that it's like a T helper or T regulatory set of cells. CD8 are found on the cytotoxic T cells. And the CD4 marker binds to and helps assist the MHC class 2 that's presenting the antigen to the T helper or T regulatory. And the CD8 marker helps to bind to the MHC class 1. So remember my little cheer: 2, 4, 1, 8. Who do we appreciate? T cells! T cells! Yay, T cells! So remember that to help you remember that the T cells, which type of T cell reacts with which type of MHC complex. So remember that the CD4 cells and the CD8 cells require an MHC marker that contains the antigen in it in order to activate. So here's the picture of that again. Here's the B cells and the T cells, and you can see the CD3 in blue and then the CD4 in brown, CD8. It could be CD4 or CD8, but you can kind of imagine this pushing over and kind of being a co-receptor if there's something here in the T cell receptor, and then this is the antigen binding site for, and then this curved part would be where the MHC2 or MHC1 marker is located, and they will kind of be upside down, fitting in this area right here. So that is a little bit more about the B cell surface markers. So T cells are going to circulate in the lymph nodes and the spleen. They're going to migrate to a specific area which we call the medulla, which is also called the paracortical region. So it is estimated that 1 billion T cells pass between the lymphatic and the general circulation each day. So imagine that this is an orange, and the outside is like the rind of the orange. We call that the cortex, and the inside fleshy part is the medulla. So when we talk about the medulla, we're talking about the center of the lymph node and the spleen. There are, and this is not just my name for it. Other textbooks do call it the cortex and medulla, but typically the outer portion, and if you think about the kidney or the adrenal gland, they have these two areas, the renal or adrenal cortex, and then they have the renal or adrenal medulla. And so it's not unheard of to call the center portion of these glandular organs the medulla, and the outer portion the cortex. So the cortex is where the follicles are located, and the paracortical, like near the cortex, is the medulla. It's not the actual cortical region or follicular region, but it is near it. So that's the medulla. So the B cells mature in the bone marrow, and that is called the bursa equivalent. The bursa equivalent, that's where we originally discovered the place where we make bone, where we make red cells, white blood cells, and platelets. We discovered this in the bursa of chicken, and so the B cells remain in the bursa equivalent, which is the bone marrow, and they don't leave to go get their receptors or to mature. So the maturation process for B cells, that's where we get the name B cells. B cells refer to the bone marrow or bursa equivalent. And there are hundreds of millions of B cells that develop there and result in the gene modification and selection. So there's this rearranging of the genes. There's some process called mRNA, alternative mRNA splicing, that gives us all these different versions of molecules, and the genes all come together in a specific way that we can then be assured that there's billions of different potential combinations, so that we know there isn't any antigen out there that the B cell hasn't made an antibody or a receptor for it. So they circulate in the blood and home into specific sites of the lymph nodes in the spleen and other lymphoid tissues where they take up residence. And again, for the B cells, this particular location, that particular lymphoid area, is called the cortical region or cortex. And also, it's a follicular area. There's follicles. And so they have immunoglobulins as their surface receptors. And so going back to that particular picture of the B cell surface, you can see the immunoglobulins have an antigen binding site here at the end that is unique and variable. And the rest of this B cell receptor is consistent with the class of antibodies that will be found here. And so all B cell receptors have the same brown area here, which is called the constant region, but this variable part is what is unique to the particular antigen that it is made for. So this particular clone or set of B cells would respond to just this antigen, the antigen that fits in this antigen binding area. And you also see the MHC2 located on the surface, so it's going to be an antigen presenting cell. In addition, because it is a nucleated cell, you should see also an MHC1 on the surface of this cell. And so it's an antigen presenting cell, that's why it has an MHC2, but it also has MHC1 on the surface. So the immunoglobulins IgD is the particular immunoglobulin, but it can be IgM as well. IgD or IgM, but this is more common as the surface receptors. So those are the immunoglobulins, the specific immunoglobulins. So Ig stands for immunoglobulin, and then D is like the Greek letter delta, and M is the Greek letter mu. And so there's specific genes that will be turned on when it's time, and they'll create these different varieties of, or classes of antibodies. And this particular one is only found, pretty much only found, it's not secreted, so it's only found on the surface. So here's some of the other differences between the B and the T cells. So the site of maturation, that is definitely one of the things that I will ask you about. Where do the B cells and T cells mature? The specific markers. You should know about the receptors for the antigens. That the B cell receptor is an immunoglobulin and the T cell receptor is a T cell receptor. The location in the lymph node, so again they call it cortex, and then paracortical sites in the follicles, interior to the follicles, so you just have to get that idea. And do they require antigen to be presented to them? And the B cells do not require it. They can receive the antigen on their own without, and they can become activated without presentation of the antigen. But the T cells do require that, except the gamma delta T cells, which can be activated differently. And then the different types of cells that are produced when they get stimulated. So when they get activated, plasma and memory cells are made. And then there's all these different classes of, or subsets of T cells that are made when the T cell gets activated. And so general functions, B cells make antibodies, and so they're the antibody mediated, and T cells make cells, so they're the cell-mediated immunity. So this table is pretty pretty much comprehensive, covering the things that I would ask you on the test about differences between B cells and T cells. So there might be a matching or a multiple choice question where you have to match, does this apply to the B cells or the T cells? And it'll be some of these distinct differences between them. Could be an essay question as well, but more than likely it'll be a matching. So each naive lymphocyte bears this antigen receptor that recognizes a unique antigen. And again, they start out as a stem cell. They become all these different white blood cell types in the bone marrow. Lymphocytes can, and there's two different cell lines, by the way, two different stem cells. So there's like the myelogenic, and then there's the lymphogenic stem cell line, and the lymphocytes are actually on a different stem cell line than granulocytes, just FYI on that. But the stem cell differentiates into the two different types, B cell and T cell, and that is the immature B and T cell. And the B cells stay in the bone marrow, T cells migrate to the thymus, and that's, again, where they get their name. And each then gets a unique antigen receptor in the bone marrow and the thymus. So this is like getting your diploma, your degree, and then they're immunocompetent, but they're naive. They have never experienced the antigen. So they migrate to secondary lymphoid tissues, like I said, the cortex and the medulla of the lymph node or the spleen. So by the time the B cells and T cells reach the lymphoid tissue, each one is equipped with to respond to a single unique antigen. So they're immunocompetent, they are naive, and they're able to do the job. They have competency, but they have never done it. They're naive. So this picture really shows what is going on, and it's pretty exactly the same as except I didn't draw a picture of a bone marrow and thymus. But they release these immature lymphocytes. They differentiate and mature into the B cell and T cell, which mature B cell and T cells will have their receptors, and then they go to the lymphoid organ, again, in the cortical region. You can see those circular areas are the cortical or follicles, and the T cells are sort of in the center of the medulla. So why can they have so many different, how can they respond to so many different varieties of antigens? Well, the variable region out here is going to pick like 4 different kind of sets of genes, and it just like randomly picks these, and that is put together, so it's spliced together uniquely. And so there are billions of trillions of possible combinations of all of these hundreds of different genes if you combine them in these regions. So that every shape imaginable of antigen there is a B cell or T cell for that. So the B cell receptor again has the 2 polysaccharide protein glycoprotein sections, and this is the variable region here, which is responding uniquely to the antigens, the unique shape that fits the particular antigen. And the heavy chains here and light chains, those are the receptors. They're also the immunoglobulin, and so a lot of this is like repetitious because it's repeated several different times in different ways. But again, the constant region is unique to the class of antibody. So like IgGs all are the same, IgMs are all the same, IgD is all the same, IgA is all the same, IgE is all the same here in the constant region. But the unique variable part is going to be found if you came from the same clone, they would all have the same variable region. So the antigen binding sites are the ends of those forks that can be highly variable, and they fit perfectly with that particular antigen that they're made for. And the variable regions are a result of genetic reassortment, so that every antigen that you can possibly imagine can be detected by some B or C T cell. The T cell receptors are formed similar, and they have the same genetic modification, variable region and constant region. that's inserted into the, but it's inserted into the membrane. It has two membrane-bound sites. Instead of just sitting on top of it, it's more embedded in the membrane, and it is relatively small, never secreted. It's not an antibody, but that is what is similar in shape in the fact that they have variable and constant regions, but they are different in shape. So the receptor, you can see, has this area for binding to the MHC with the antigen in it. And so again, two submembrane, where the B cells, they kind of sit in the membrane. This goes through the membrane, transmembrane. So it's just slightly different attachment here. So clonal selection is when you have all this repertoire from the lymphocytic stem cell. You have all these different B cells and T cells now that have been made and have all unique antigens that they're going to respond to and be activated by. Well, during this process called clonal deletion, we create some immune tolerance to the self-antigens. We do not want to respond to self-antigens. If we do, then we're attacking ourselves, and so that's not good. So this is when you see the repertoire that's left, those are ones that do not respond to self-antigens. And when you talk about clonal selection, that's when one entry of one epitope, one antigen, binds specifically to this one clone, and it makes then copies of it. It proliferates and differentiates, which is kind of a good transition into the third step here. So this is actually when you have antigen presentation. So this is development, antigen presentation, and then you have the activation or challenge of the B and T cells. So immune tolerance is a tolerance to self-antigens, so it can remove potentially harmful clones through clonal deletion, and it's thought that the autoimmune diseases would have been caused by a loss of immune tolerance, the survival of some forbidden clones, or failure of other systems. So there's a lot of different ideas, but that makes sense. That particular theory does make a lot of sense, and there's a lot of information and research to back that. All right, so concept check 2. The T cells are display the CD3 marker in either a blank or blank co-receptor. So when we were talking about co-receptors, that's the CD4 or the CD8 co-receptor, depending on the cell type they are. The B cell receptor is a blank molecule. So I think we mentioned it as a glycoprotein. And true or false, the T cell receptors are similar to B cell receptors, except they're not secreted. So that is true. They are not secreted, and they are made in a similar way. They have a variable and a constant region. True or false, the lymphocyte specificity is pre-programmed. I guess it depends on how you define pre-programmed. But yes, in the genes, there are all these different possible shapes of the variable regions, and then this reassortment of the genes is what gives them their specificity to the antigens. So true or false, B and T cells never react to self-antigens. So if you answered that is false, you are on the right, you are correct. So whenever you see never in a true or false question, that's usually meaning that it is false, because very rarely can we say something is always or never something. So that is false. B cells and T cells, we delete the clones, the forbidden clones, but sometimes they don't get deleted, and that's why we do have some autoimmune diseases. So that's the end of section 2.